Alrighty. Thank you all for coming out. I'm Brian Lerch, and today I'm going to be talking about a series of projects that will help us understand the evolution of same-sex sexual behavior. So we'll start with the fact that same-sex sexual behavior, which I'll refer to as SSB, is extremely widespread. It occurs in a wide range of taxa, including taxa that are quite unrelated. Despite the fact it's so widespread, it's been fairly neglected evolutionarily from studies, and I think a big reason for this neglect is due to the perspective that researchers have tended to take. Researchers are quick to point out that SSB doesn't result in reproduction. It's often referred to as an evolutionary paradox. The fitness costs are stressed. And what we're really going to do for the rest of this talk is kind of unpack this perspective and kind of critically assess whether it's the right way to be thinking about SSB. So to do this, we'll just start off with a really simple mathematical model on the evolution of sex discrimination. So we'll model two sexes. The first one we'll refer to as the searching sex, the second one the targeted sex. The searching sex is capable of discriminating the sex of conspecifics. It might bias mating attempts towards the opposite sex. We'll call this discriminant mating. And we'll assume that this sex discrimination carries some cost. You can think of this like a search cost. On the other hand, like that searcher with the white S, the searching sex might mate indiscriminately, in which case it'll put equal mating effort with any conspecifics. The targeted sex, on the other hand, may or may not provide some signal to identify themselves as targets. In cases where the target does not signal, like the blue one up top, searchers that are attempting to discriminate will not be able to bias matings towards those non-signaling targets. And again, we'll make the assumption that signaling is costly. So the first thing that we can look at, this is a really simple model. We can treat it analytically and assess what's the optimal strategy for sex discrimination. We're going to look at this across different discrimination costs on the horizontal and different signal strengths on the vertical. Darker colors will be weaker sex discrimination. And what you see is that under a wide range of conditions, indiscriminate mating is the optimal strategy. It doesn't take very strong discrimination costs necessarily to get imperfect or sex discrimination or indiscriminate mating. And a consequence of this is that as a part of this optimal strategy, you can get quite a bit of SSB occurring. In cases where we've got black there on the right graph, that's where the amount of SSB is just given by the sex ratio itself. Even in cases where you get really strong sex discrimination evolving, you might see quite a bit of SSB just as a consequence of that imperfect signal by the targets. So, okay, this is nice kind of proof of concept, right, that you can get the evolution of indiscriminate mating, but that's a really simple model, and one of the key assumptions that we made was that those sexual signals were fixed. You could imagine if those signals are able to evolve freely, kind of co-evolve, maybe there's some positive feedback that's going to take you out of having indiscriminate mating as the evolved strategy. So to assess this, we can really just use the same model, but also let those signals evolve. So this is across a range of initial conditions. Initial frequency of discriminators is on the horizontal. So this is binary. Each individual either discriminates or doesn't. Over on the right, that's where all searchers discriminate. Over on the left, that's where no searchers discriminate. The vertical axis then is the initial frequency of signalers. Again, it's binary. You signal or you don't. Up top, every target is signaling. Down on the bottom, no targets are signaling. So when we look across this range of initial conditions, we see what the different evolutionary outcomes are, and we see two possibilities. In cases where you have sufficiently many discriminators and signalers initially, you get the evolution of strong sexual signals and strong sex discrimination. On the other hand, if you have sufficiently few discriminators and signalers initially, then you get the evolution of no signals and indiscriminate mating. And it's worth noting you can actually prove analytically that basically this green region always exists. Indiscriminate mating and no signaling is always a stable equilibrium, which means that if we start close enough to that case where we have few signalers and few discriminators, we'll always end up at this equilibrium with indiscriminate mating and no signaling. We think this has some interesting implications for thinking about ancestral states. So of course, sex discrimination has evolved in many cases. So what this tells us is that and other researchers have argued, and we agree, that indiscriminate mating is likely ancestral. So what that means is, in order to get some sex discrimination evolving, some detectable differences between the sexes must arise as a byproduct of the evolution of two sexes in the first place. Otherwise, we wouldn't expect to see sex discrimination evolving. So these first two parts of the talk kind of go to show that indiscriminate mating should be expected to evolve, and thus it doesn't really make sense to view SSB as a paradox, but rather SSB is really something we should expect to see in systems with sexual reproduction. But as I already pointed out, those are really simple models, pretty abstract models, and what we want to do is kind of think about whether we can actually use this theory, develop some theory that we can connect to empirical studies or to some of the things that empiricists who are working on SSB are actually observing and measuring. So that's what the next part of the talk is going to do, kind of shift gears and think about some of those mechanisms. So the first mechanism we're going to consider is learned sex discrimination. And to motivate this part of the talk, we'll note that those past models suggest that male bias sex ratio should lead to the evolution of stronger sex discrimination in males. What's interesting is that a number of empirical studies that consider plastic or learned sex discrimination show something different. They either find that male bias sex ratios lead to weaker sex discrimination, or maybe there's no influence of sex ratio. So the primary question of this part of the talk is basically, why is there this disconnect between what we see with innate sex discrimination versus learned sex discrimination? And I should note that there's I know of one example of experimental evolution that agrees with theory that suggests that evolved sex discrimination should have stronger male sex discrimination in male bias sex ratios. So to develop this model, it'll be fairly simple to what we talked about before. Now, matching those empirical studies, we'll explicitly treat the searching sex as males, the targeted sex as females. The first step is that males can attempt some mating based on innate sex discrimination. And then after mating, males might update their sex discrimination strategy based on learning. After they've learned some further sex discrimination, males will attempt further matings with that learned sex discrimination, and those steps of learning and mating can repeat multiple times. We'll consider three strategies for how learning can work in the model. The first strategy we'll call learn from males, which is where males will only learn from unsuccessful mating attempts with other males. The second strategy, learn from females, males will only learn from successful mating attempts with females. And finally, learn from both, males learn from all mating attempts. So the first thing we want to do is characterize how these different learning strategies interact with the sex ratio. So horizontal axis here is basically the sex ratio. What's the proportion of females you interact with early in life? Over on the right, that's all female. Over on the left, that's all male. Vertical axis here is a measure of sex discrimination, stronger up top. And I'll note that that measure of sex discrimination is under common conditions later in life. And what you see is that these different learning strategies clearly lead to different influences of the sex ratio. And it makes a lot of sense. We'll take learn from males just as one example. If you only learn from attempting to mate with males, then it's under those conditions that you experience a lot of males that you'll get strong sex discrimination. If you're only experiencing females but you only learn from males, you won't learn sex discrimination. Learn from females is the opposite. So now what we're going to do is compete those different strategies against each other evolutionarily and ask what learning strategy should evolve. And what these graphs show are three different parameters: death probability, cost of discrimination, and maximum number of mating attempts, and then the probability that different learning strategies fix. And we see these graphs are mostly blue, indicating learn from males is the strategy that most often evolves. This should make sense because, as I've already told you, theory suggests that male bias sex ratios should favor stronger sex discrimination in males. Learn from males, therefore, leads to an adaptive norm of reaction if you look across different possible sex ratios. But it's worth noting that some of the conditions disfavor learn from males or favor some of the other strategies. So, for example, high death probabilities, low cost of discrimination, and few possible mating attempts are all going to favor learn from both, in part because learn from both leads to the strongest possible sex discrimination, which is what you might expect to be beneficial under those three conditions. And it's worth noting that those conditions might also be common in the insect systems that have been studied empirically, right? Insects tend to have pretty short lives. Those are going to constrain the number of mating attempts that they might have. And these costs of discrimination really haven't been documented, or at least not been documented as strong in insects. So this suggests that learned sex discrimination might be one mechanism that has some empirical support that might be accounting for SSB. And this disconnect between the role of the sex ratio in past empirical and theoretical studies might just be kind of an artifact of insect life history, which is the taxa that's been studied the most extensively empirically. So now I'm going to shift gears one final time and talk about another project that's going to try to make some empirical connections or consider some mechanisms that are really observed. And this is based on the observation that in a number of systems there might be a trade-off between the total number of matings an individual partakes in and the sex discrimination. So this model is going to be a little different than past ones we've talked about. We'll have individuals randomly moving through space, and each individual is going to have what we'll call a masculinity score, sort of how much like a male do you look. When a male bumps into a conspecific, perceives a conspecific, they'll decide whether to mate based on that masculinity score. So the lower the masculinity score, the more likely a male is to decide to mate. And then the stronger sex discrimination is, the faster that probability of mating drops off as a function of masculinity. With no discrimination, a male will mate with any conspecific they bump into. And it's worth noting that this model produces this trade-off between mating rate and sex discrimination. We didn't assume this going in, but this sort of emerges from the assumptions of how mating decisions are made. But now that we've generated this trade-off, we can assess what its implications are for the evolution of sex discrimination. So first, let's consider the case where only male sex discrimination evolves. Under these assumptions, faster encounter rate leads to stronger sex discrimination. This is because at low encounter rates, the opportunity costs of mismatings are very high, so that trade-off is balanced by investing in the mating rate. Something interesting happens, however, if females are able to co-evolve, specifically assuming that high mating rate is harmful for females. In this case, female masculinity will evolve upwards females will evolve to look more like males, basically. This is in order to reduce the mating rate. As a result of that, sex discrimination becomes less effective. Sex discrimination will weaken. So as a result of male harm, there's this kind of cascading effect where females will evolve to disguise their sex, and this ultimately leads to more male SSB. So just to kind of sum up, I hopefully have convinced you that we shouldn't be thinking of SSB as a paradox, but there's a lot of mechanisms that can lead to its evolution. I'd like to thank two people in particular, Maria Servideo, who worked with me on the first three projects, Ewan Flinham, who worked on the fourth project. I'll flash up those conclusions, and I'll take any questions with what time I have left.
Can you hear me?
Yes. Yeah.
Look, and I was just wondering, like, could you speculate, like, would it be prudent to assume or expect that there would be more, like, males would discriminate less or mate more with males, given that females also look like, or would they tend to evolve even better discrimination mechanisms?
So I think one of the kind of keys of the model is that sometimes you just can't. And I think this is probably true in nature, right? Like, sometimes that mimicry might just be so good that there's nothing that can be done about it. And in that case, then I think the theory would suggest that you want to invest in increasing the mating rate at the expense of sex discrimination. Thank you.
All right. Hi, my name is Jay Bundy, and boy, am I wishing I paid more attention in French class. This coming fall, I'll be starting as an assistant professor at Seton Hall University in the Department of Biological Sciences. But the research I'll be presenting here actually began at the University of Minnesota in the Travisano Lab, so special shout out to the Travisano Lab. It's called Specialization, Pressure, and Resource Limitation as Drivers of Mating Type Differentiation and Sexual Selection in Digital Organisms. I know it's a mouthful, but we'll get there. All right, so this work is about artificial life, which for my purposes is a branch of biology. We focus on the use of data and algorithms to evolve organisms with brains that solve problems. In particular, I'll be talking about experiments with the digital organisms of MABE, which stands for Modular Agent-Based Evolver. Basically, these are digital organisms with genomes that encode brains. They also encode an agent, so any thing that may be considered the body or whatever of the physical agent. They are also internal container classes of the optimizer and the archivist that are used for sort of evaluating the organisms and storing their data. And the organisms exist in a world. For our purposes, a brain is considered a controller, a solver, a solution, or even a machine. In MABE, we can plug and play different brains because they're different modules. But I'll primarily be focusing on results that we get with CGP, Cartesian Genetic Programming brains, a little bit with Markov brains, and I'll mention briefly recurring neural nets. So one of our driving questions is, why do different brains evolve different behavior? Something I'm not going to spend a whole lot of time on today, but that is one of the large questions that we are looking for in this system. And so we'll begin by talking about a game. And those of you that are familiar with machine learning, this type of research commonly uses games, checkers, chess, Go. But for our purposes today, we're going to be talking about a mating game. And our mating game actually does start with a grid-based game, but instead of being checkers or chess, we call this Berry World. And you can think of a simple version of Berry World where each location on the grid contains either a red or blue berry. We drop the organisms in randomly, and they can evolve the ability to move around and eat and things like that. And so if they eat a berry, they get 1 point. If they eat a different colored berry, that's a switch cost, they will get a penalty of 1.5 points. And all berries that they eat are replaced randomly. So if they eat a lot of red berries, there'll be a lot of blueberries, and vice versa. And so one of the things that's very interesting is to pick a winner. We start with a tournament, and it's a gauntlet-style tournament. So we basically take two organisms, we figure out which one has the higher score based on their performance in Berry World, and they go to the next round, we pick another winner, and so on through about 8 rounds. And then congratulations, we have a winner, you're done adulting, and you can go to Disney World. But not so fast. You're actually going to the mating chamber, and before you get to go have your fun, you need to find a mate. And we all know things get wonky when you've got to find a mate. So we have a bird here saying, how do you want to go out sometime? Sorry, no, I'm only into large ovals with screaming neon mouths. Of course, the large oval with a screaming neon mouth shows up, and off she goes. So in our case, we need to build a lek. We've got a winner, and now we need to build a lek so that winner can find a mate. This lek I'm showing here is basically just like you can think of as an enclosed arena where potential mates compete. This one is showing males having territories of different sizes. So A has the biggest territory size, B is the next biggest, and so on down to D being males with the smallest territory size. But you don't care about territory size, you care about color. And generally that means you prefer red or blue mates. So to find you a mate, we're going to build the lek, and we do that by having another tournament, except this tournament is winners only. So you have to win a tournament to get into the lek, and then based on your color, like if I prefer red, I'm gonna choose the reddest organism that made it through into the lek. Of course, because you care about brains too, and so we've got to have some good old natural selection in there. All right, now you're done, and congrats, you finally found a mate, and it's time to make an offspring. But not so fast, because what kind of game show would this be without some vile twist at the end? And so in this system, your reproductive chances are determined by your body shape. And in this case, organisms' body shape is determined by a single number between +1 and -1. If it's -1, it basically means your body's perfectly concave. +1 would mean you're perfectly convex. And you actually mate best based on fit, right? So +1 and -1 actually does really well together, but +1 and +1 is no good, and neither is -1 and -1. And we call this system shape mating. And so one of the things that's really interesting is you can see this function that we use, and it's selecting for maximally different shapes, but there is an exponent on this function, and when you look at it with the exponent being 1, you see these nice little quadrants, and so we consider the anisogamy pressure in this case rather weak, and in most of the time when we use an exponent of 1, the organisms all evolve to that intermediate shape of basically 0, and then they have inefficient mating, but it's like good enough for them. If we make the exponent 2 and we make that shape mating function far more severe, they almost always do shape mating, and they evolve to either be -1 or +1. So this leads to like a 3-stage game. First, you've got to harvest food to be good enough to win the tournament and be the first parent, or get into the lek to potentially be the second parent. You then find a mate based on your color preference, and then finally your chances of reproducing in the mating chamber are determined by your shape. And so organisms develop a number of really interesting strategies to deal with this, but the one we're going to be focusing on today is cross-signaling. And cross-signaling is basically the idea that you become one extreme shape or the other, you then eat one particular food or the other, and then you mate with the opposite shape that eats the opposite food. And it's beautiful. It works really well. But a couple interesting things. So first, like I mentioned, if we don't have strong anisogamy pressure, we don't get the 2 shapes. So that's condition 1. We got to kind of crank that exponent up to 2 or higher to get them to reliably do shape mating. And then without that switch cost, they don't do signaling. They'll eat both foods, and then they'll eat both colors, and when they go to pick somebody of the right color, they often get the wrong shape. So that's why we talk about anisogamy pressure, and then we talk about specialization pressure being that switch cost. And so this is what it looks like when cross-signaling doesn't evolve. So if they don't have the switch cost, this is what I was talking about before, you can see that these organisms are eating both red and blue foods, and we have red and blue eaters in both shapes, and they prefer the same color, not the opposite color, and so their mating is really inefficient. All right, and then we can sort of start to look through the various brain types we have and see how they perform with this. So our CGP brains, we do runs about 10,000 generations, and they almost always find the cross-signaling strategy. They love it. They're really good at it. They get an A+. The Markov brains are a little bit less efficient. They only do it, say, 32 out of 50 times, but we're using a university scaling system, so that's still an F. And this is what they look like without cross-signaling. So you can see again that they're eating both colors in each shape, and they have inefficient mating. And then the RNNs, they only do it about half-time, but I say see me after class because they do this really interesting thing where they actually do distinguish their diets and their shapes. They just don't do the signaling thing on top of it, whereas with the other 2 brain types, if they distinguish their diets, they always find the signaling strategy. So we actually see like sort of bits and pieces of the strategy come together in different brain types. And one of the things that we've discovered is that we can sort of change the way mutations are handled, for instance, with the Markov brains, and make the genetic elements more mobile, and they'll actually behave more like CGPs. So that's just a sort of interesting side note. One of the other interesting things we get, and we're focusing mostly on the CGP results here because they're the best performers, is they do this cross-signaling with 3 food types, where there's pluses and minuses. They eat one color, they prefer the other color, but since there's 3 food types, right, they want somebody to be eating that third food too. And so they develop like these 4 mating types where the third color, and here being green, that would be the third food, there's like pluses and minuses that prefer the correct opposite shape. And they do this a lot, and it actually forms a really unique cycle. So with 2 food types, they're stable, but with 3, they do these cycles where, like in this case, they're eating a lot of blue and red. So the next one, there'll be a lot of green, and so those green guys kind of pop up, and then they'll pop down. It's actually pretty cool. And then with 4 food types, we get this other really cool thing that they do, where you have red eaters who prefer purple, green eaters who prefer blue, purple eaters who prefer green, and then the blues prefer red. So so you have 4 different mating types, but they're crossed. All of their preferences are unreciprocated. And this becomes really stable because they're eating all of the foods, but they're also perfectly choosing mates of the opposite shape. So it's just a pretty interesting system. We didn't know if they would do anything interesting. We just kind of wanted to see if we could get shape mating working, and we found some really interesting results. So to summarize them, shape mating doesn't evolve without sufficient anisogamy pressure. Signaling doesn't typically evolve without switch cost. Different brain architectures produce different strategies, and the number of food types influences the number of distinct mating types. All right. Thank you. Go Pirates. And I think I have some time for questions. Questions? Yeah.
Thank you. That's the first time I've ever been introduced to the MABE system. It's really interesting. I'm wondering, I might have missed it. So can you just, if you don't mind, talking a little more about how the offspring are produced and what traits they end up with?
Yeah, so we go through and we evaluate all of the organisms basically, and then we put all their scores into a list, and then we go through this optimization process of picking new parents. So like I talked about, you win a tournament, that gives us one parent, that parent chooses elect, and they go through another parent. They'll make an offspring, that offspring gets put into a list of the new population, and then we just delete all of the parents.
How do you decide what traits the offspring has based on the 2 parents?
Oh, their genomes. They do like crossover at a random point, and they have a configuration file where you can set different mutation rates. Yeah, so I didn't go a lot into the genomics of it, but yeah, they just have string genomes that are usually numbers between like 0 and 255, and then they're just subject to random mutations. And we can program mutation types, so if you want to add insertions, deletions, whether they can shift them, invert them. There's a lot of stuff like that. But these ones are using really simple, just string genomes. Yeah.
Mutations don't affect the fundamental architecture of the brain.
So that's one of the different things. So for instance, with like the CGPs, all of their business logic ends up being really close. Whereas like a Markov brain by nature distributes its logic throughout the genome. And so that was one of those things we discovered where if we made the Markov brains more like the CGPs, we actually could change their behavior. So there is a little bit of a difference in terms of how the particular brains are encoded by the genomes. And I can definitely, tell you more about that. And I have these files. They're really simple to play around with if you're interested.
Super cool topic.
Thank you.
So is the tournament like a single elimination?
Yes, but I call it gauntlet style, right? Because it's not like brackets and then winners go on. It's just you choose one and the winner stays. Pick another one and the winner stays. Choose one and the winner stays. Yeah.
So it's almost kind of like a round robin.
In a little bit, yeah. But there's only 8 of them that it goes through. So, yeah. Any other questions? All right. Thank you all so much. I really appreciate your time and being here today.
Thanks, everyone, for coming. It's pretty late, so I appreciate you staying to hear me. My name's Andrew. I'm in the last year of my PhD, and I'm really excited to share with you some preliminary data for my last chapters. The light is at the end of the tunnel for my graduation, where I am testing the genic capture hypothesis in a really cool species of Lecking prairie grouse, which I'll get into in a second. And so just for a broad overview of things, Lecking has always kind of been an evolutionary puzzle for a lot of biologists. So Lecking with this large congregation of males where they're all performing these elaborate displays in hopes of attracting a female would presumably lead to the selection of a single or very few optimal phenotypes. And so that's expected to erode genetic variation. So you have this positive directional selection for an optimal male phenotype, which is expected. However, genetic variation for a lot of these traits and genetic variation overall in Lecking systems still persists, which a lot of biologists have termed the Lech paradox. And so I became really interested in this question of the Lech paradox and how these organisms may maintain sufficient levels of variation to persist across evolutionary time. And so one of the potential hypotheses that has been proposed is called genic capture. And so in essence, genic capture is explaining that the traits that an organism expresses are reflective of the individual's condition. And the condition is defined as the pool of genome-wide variants that contribute to fitness. So it's not one particular region of the genome that's under selection, but it's a wide array of variants that are scattered across the genome that all collectively contribute to fitness. And so Rowe and Houle in 1996 published this theoretical model to explain this. And so the figure on the left shows that the condition, or the genome-wide variance, as that varies for each individual, the trait value will vary. And what this mathematically translates into is that the mean condition of a population, as that increases, the genetic diversity of a trait will also increase. And so this is what the authors term as condition dependence. And so the traits have to be an honest signal of the organism's condition or the genome-wide variance. Kind of on the converse, I wanted to show the mathematical illustration of what no condition dependence looks like, right? So as the condition varies, the genome-wide variants vary. The trait does not change, so it's not an honest signal. And as you can see, it preserves much lower genetic diversity than we observed in the previous example. So it still does change a little bit, but overall, much lower variation is preserved. And one of the key take-home messages is that the genic capture hypothesis is unsupported without condition dependence. So if we don't have those honest signals of condition, the genic capture hypothesis fails. And so this was published theoretically several decades ago. And so what does this look like empirically? And so there's been relatively few empirical tests, and the tests that have been done so far have been done in laboratory model organisms. And so the most straightforward one that I really like to use as an example here was published in 2019 by Dugand et al. In Nature Communications, where they manipulated the mating success of Drosophila. And to synthesize their results, on the x-axis here is mating success. So as you move forward, you increase your mating success. And then on the y-axis, you have heterozygosity, a measurement of genetic variation. And so if you look specifically at phenotype-associated genomic regions, so regions of the genome that are specifically related to the phenotype, you have a reduction in heterozygosity as male mating success increases. Now, when you look at the entire genome, maybe less so, but you still see that same pattern. And again, that's because it is a condition-dependent trait, and so the genome-wide variants are all effectively under selection. And so this is what we would expect genic capture to look like in an empirical system. But unfortunately, it's been really hard to test that in a wild system until now. And so part of my dissertation is to test this hypothesis in a really charismatic bird called the lesser prairie chicken. And so the lesser prairie chicken is native to basically the middle of the United States. So this is a map of their range. They form two distinct population segments, the southern one in Texas and New Mexico, and the northern one in Oklahoma, Kansas, part of Colorado. And what the males will do is they'll congregate on these leks in the field and perform really quirky, wonky behaviors where they'll jump around, they'll inflate their sacks, they'll make all sorts of vocalizations, bob their heads, The females are in the periphery watching these males, finding the most attractive one for them to mate with. And you can see some traps as researchers were sampling these species. And so for this study, in order to test genic capture, we took whole genome resequencing of about 248 lesser prairie chickens, which included 166 males, 82 females. We measured 8 different phenotypic traits, which are listed here, that are known to influence male mating success. And so with these 8 traits, we evaluated them individually as part of the condition, and then we also distilled them together to their principal components to explain the variation across all 8 traits. And so to define what we mean by condition as it applies to our study system, we decided to take 2 metrics in an attempt to kind of replicate the experimental results from the Drosophila experiment. And so big H is what we designate as overall genomic condition, which is just the measure of genome-wide heterozygosity, or the proportion of heterozygote sites scattered across the genome. Now, little h sub I is the genetic diversity of a particular trait. And so this is measured as the amount of heterozygote sites at the top 10 windows across the genome that are associated with a specific trait. And in order to do this, we ran a genome-wide association study. We averaged the association values of our single nucleotide polymorphisms across 50 kb windows, and then took the top 10 windows, filtered our files down to those to measure heterozygosity at those very restricted windows. And so that's what this is showing. And so I want to get into the results here. I am only focusing on males today, but if you want to talk about females, come see me afterward. And so this is the principal component analysis of our male traits. PC1 and PC2 explain roughly 42% of the variation, and as you can see, there's no real distinct clusters, but the traits load differently on each principal component axis. And what we expected was that there would be different peaks of association, so different windows that are associated with each principal component axis. That's exactly what we found. And so, like I said, again, we took those top 10, which are approximately at that region of the median likelihood ratio statistic of those genomic windows, and we calculated heterozygosity. And so the figures on the left here are the principal component scores of each PC axis, and then on the y-axis is genome-wide heterozygosity. And as you can see, there is no relationship between genome-wide heterozygosity and the score of each principal component axis. But what was really interesting as we dove deeper into these specific genomic windows, PC1 showed a negative relationship with the principal component score and the heterozygosity present only at those windows, which is indicative of some type of directional selection for phenotypic variability in those males. A much weaker relationship exists for PC2, but that explained much less of the variation, and all of the traits loaded positively on that, so that wasn't entirely unexpected. When we look at individual traits, for all 8 traits, unfortunately, big H, or genome-wide heterozygosity, shows no condition dependence. It's that flat, unassociated line. But what was interesting, again, focusing on those specific genomic windows, is that heterozygosity for wing and heterozygosity for the comb area showed condition dependence. Wing specifically showed a positive relationship, so as wing size increases, heterozygosity at those windows would increase. We observed the opposite pattern for comb area. And that's really interesting because wing size and comb area are traits that influence how successful a male is on the lek at attracting and mating with females. So to kind of summarize, this is what we expect under genic capture for condition dependence, a high correlation between the condition or genome-wide diversity and the trait value. And I would argue that our study kind of shows low condition dependence. If we focus on specific regions of the genome, we do identify condition dependence, but across the entire genome, we really don't identify that pattern. And so in summary, the phenotypes important for sexual selection in the lesser prairie chicken do not reflect the overall condition of the individual, which is indicated by the lack of a correlation with big H and any phenotype. We thus fail to support the gene capture hypothesis in the lesser prairie chicken lecking system. However, we argue that there is evidence of potential directional selection for these various phenotypes that I pointed out, indicated by a significant correlation. And to kind of bring it back to the diagram for the Drosophila literature, remember we should have seen, if gene capture were supported, a negative relationship between a trait or mating success and heterozygosity. We do see that in some of our traits specifically, but we fail to provide that evidence genome-wide. So unfortunately, there's no good answer. This was, really kind of a thing. I knew I was jumping into something that was quite messy. Like paradox has been unexplained for quite some time. But it also opens new avenues of research. We have such great genomic data available to us. And researchers do also collect a lot of different phenotypic traits depending on what their organism is. And so for future tests of the gene capture hypothesis, researchers should really prioritize first the identification of condition-dependent traits, if that is possible in your study organism, because those traits would be good candidates to assess this hypothesis. Ongoing work is currently assessing if other metrics of genomic diversity, including Watterson's theta or nucleotide diversity, may be a better indicator of condition than heterozygosity. Heterozygosity is influenced by demography and a whole bunch of other different things, and so we're going to compare that with the other measurements. If anyone's coming to the Midwest Population Genetics meeting in Indiana University in 2 weeks, maybe I'll show you some results for that. That's what I have for today. I would love to chat with people about this. If you have any ideas, I'd like to acknowledge 2 funding sources, Western Association of Fish and Wildlife Agencies, NSF. The genomic and phenotypic data was a large collaborative effort. A lot of people were involved in collecting those data, so thank you to all of those. And then my very supportive labs are wonderful to work with these last 5 years. And so here's my information. I'll take questions.
So if I caught them all, the phenotypic traits that you measured were all physical traits?
Correct.
But in fact, probably to prairie chickens, there's maybe the behavioral traits, aspects of the display might be important and might reflect condition as well.
Yeah, that's true. If I had been collecting the data, I was not involved with that data collection, but I would have loved to basically evaluate behaviors. They know the behavioral literature suggests that a lack of latency, so as long as the male is a little more active on the lek, that influences his mating success. I would love to assess heritability of a lack of latency and see how that plays into it too. Unfortunately, we don't have that data. Yeah. Okay, thank you very much. I'll be outside too if anyone has any other questions.